Okay, Assalamualaikum and a very good morning to everyone. Welcome to today's breakfast at UMHealth seminar, and today we will be talking about Central Research Laboratories, or in short CRL. I am Shatrah Othman. I am currently the acting head of CRL, and today I will share with you the facilities and services that we have at CRL. Especially to those who are not familiar with our facilities. Next, we will then invite two of our users to share their experience on using our facilities. Okay, so next, let me share my slides. Okay, so this is CRL. And CRL, or stands for Central Research Laboratories, consists of MBL, TCL, CMBL, FOM supply store, and FOMCAL. So these might just sound like initials to you, but I will explain to you later what they stand for. Just a quick history of CRL. We initiated we were first established as Medical Biotechnology Laboratory back in 2005. And during the HIR era, we managed to procure many cutting-edge technologies or instruments. And in 2014, Central Research Laboratories was established as an umbrella unit to consist of all the laboratories under it. And in 2017, MBL was rebranded to Datuk Mahmud American Medical Biotechnology Lab. Okay, so this is our vision and mission. In terms of organization, CRL sits under the Dean of Faculty of Medicine, Professor Dr. April Kamila Roslani, and the Deputy Dean of Research, Professor Dr. Sanjay Rampal. There are four, let's see, why is my mouse not moving? Okay, so there are four core units under this. The biggest is MBL, okay, Medical Biotechnology Laboratory. We also have the Cell and Molecular Biology Lab, CMBL, Translational Core Lab, TCL, and the FOM Supply Store. These are the management team for CRL. Okay, we have Associate Professor Dr. Putri Shafinas Akma, who is the head. I am the deputy head, and we have Associate Professor Dr. Ivy Chung, who manages the TCL and the FOM Supply Store, and Dr. Hui Mili, who is managing the CMBL. We have two science officers, Ms. Tibashini and Puan Melissa, who are managing the entire CL as well. Just to share with you some familiar faces of the technical staff at CRL. So if you have visited us or you have used our facilities, some of these faces may be familiar, and they are in charge of each and every unit, every equipment that we have at CRL. On the functional side, the 12 support staff, or the technical staff, and the two science officers are also involved in the laboratory facilities, including all the different parts of the labs, the different services that we provide, and also the FOM supply store. So just I'm just showing you this chart so that you know who is in charge of which equipment should you need to inquire later on. Okay, so the first and the biggest arm of CRL is the MBL, or Datuk Mahmud American Medical Biotechnology Laboratory. This is located at Block B, 4th floor of FOM. It's just opposite the center point, if you are familiar with center point. It comprises of the proteomics facility. Okay, so we have the huge lab for proteomics, and we have the genomics, and also the FOMCAL here. We have many lab benches and workstations. We have a mini lecture theatre that can fit up to 50 people. So this is also available for rental. or for FOM users, you can book this for your events as well. So these facilities will is housing all the high tech and even the basic equipments, which I will share with you later on. So just to share with you what it looks like for those who have not been to our facility. So if as soon as you enter the CRL at this door here, to the right is the proteomics facility. And once you go through the door there and across to the other side of the block, you will get to the genomics facility. So this is what it looks like. Don't be too bothered about how empty it may look now, because we just don't want to put pictures with too many people that might clutter the looks. Okay, so next is the Translational Core Lab, TCL. This is actually located on the UMMC side. Okay, it's on the Complex Kesihatan Wanita dan Kanak-kanak level 11. The target users for this lab is actually the clinicians who have projects that has to do with molecular and cellular research. So there are many facilities, including cell culture suites and lab benches that can be used by the users. Okay, this is what it looks like. Okay. These are the labs, some of the equipments available, and the cell culture or laminar flow. Next is the Cell and Molecular Biology Lab, or in short, we call this CMBL. This is located at Block T, Level 3 FOM. Okay, from the inside, it might not be too visible, but if you look from outside, this building here may be more familiar to you. What we do here is actually to provide facilities mainly on cell culture, okay, and all the molecular biology equipments that you need for that. Okay, so the whole range is also there: the real-time PCR, incubator, laminar flow, microscope, and all that. Let me show you what it looks like. So this is it. So there are many lab benches. We also have workstations. We have cell culture hoods. So this is actually targeted towards researchers who have or new lecturers maybe who do not have their own labs yet. So you can rent out space at our CMBL, or you can get your students to come and perform their work here. Next is the FOM calibration laboratory. This was actually initiated in 2016, and the main purpose for this was to provide calibration service to FOM researchers. And why this was established was that by providing this service, we can have the calibration done faster, cheaper, and by a certified calibrator. Okay, so you don't have to send your pipetters or your weighing scale to an external party anymore. You can always send it to us so that you can have the pipetters back faster. Next is the FOM supply store. Okay, so this yellow building here is very catchy, so you might have already seen it. It sits right next to the center point at level 4 FOM. This is actually a one-stop center where we sell consumables and reagent kits. Okay, however, you can always purchase the products via online. Okay, so if you go to, or if you Google ResearchCard 360, it will get you to our online store, and all our products are displayed there. You can order online, and these are the different modes of payments that you can do. Okay, so in terms of facilities, okay, so I'm going to go through some of the equipments that we have. For proteomics, we have the whole range. Okay, we have starting from the vacuum concentrator for your proteins, the immobilization equipment, the first D apparatus, the Ruby set for the 2D, and we have the imaging system for the spots, and the software to analyze the spots as well. So on the high end, we have the Multitof, we have the QTOF, and we have the Bico as well. These are all available for service. Okay, next is the genomics facility. So even for genomics also, we have the whole range from the basic thermocycler or PCR machine to the higher end real-time PCR machine. We have the nanodrop for quantification of DNA and RNA. We have cryotome for histological slides prep, and we have the fluorescence microscope for visualization of your results. For genomics also, we have high-end equipments, and this includes the flow cytometry for cell analysis. We have the cell sorter, even higher end. And we also have real-time cell analyzer for analyzing live cells. Okay, so if you're doing cell culture and this is something that might interest you. We also have a high-end plate reader, okay, that can be used using multiple modes. On the cell culture side, the CMBL houses several units of laminar flow, and it also has the microscopes and the incubators. And on top of the normal incubator, we also have the hypoxic incubator, which may interest some of the researchers who are working under hypoxic, cells under hypoxic conditions. For sample storage, we have the whole range of freezers and chillers, okay, from the 4 degrees right up to 150 degrees. So, and the space here is available for rental. You can either rent the whole shelf or buy boxes, depending on how much samples you need to store. So do approach us if you do not have your own -80, for example. This can be rented. These are some of the, just to show you some pictures of the bench space that we have. So this is at CMBL. These are some of the workstations that we rent out as well on monthly basis. So do approach us if you need to rent some of the spaces, maybe for your postdoc or your visiting lecturers or whatever. So this is what it looks like at MBL, and these are the workstations at CMBL. On the services side, we have several services. So if you have any interest in any of these, do approach us. So we have protein digestion, profiling for proteomics and metabolomics. We have functional cell analysis, and right down to the basic cell culture, okay, and proliferation assay. These are ongoing throughout the year. Okay, so if you have interest in other than the assays that I've mentioned before, or the services that we mentioned before, you can also approach us and we try and customize the service for you. We also do lots of trainings throughout the year. Okay, so just watch out for the announcements on the UM info email and FOM emails. From this ranges from workshops on cell culture and proteomics, and even hands-on or operator training workshops for specialized equipment such as the FACSCANTO or real-time PCR. So if you have, if you're a new user and you want to use this equipment and you want to have hands-on on this, we can train you. For the upcoming year, we also have plans for internship programs. Okay, this is tailoring towards interns, maybe even from outside the university, to expose them to give them better exposure to the laboratories, to the equipments, before they venture out into the job market. We also have induction program. So this is targeted towards new students or new RAs, so that they are more familiar with the lab that we have. And also, if you need us to train you in certain equipments, we can also do that for you. These are just to show you some of our collaborators. Okay, we have Agilent, Cancer Research, Paramanantham, Research Instruments, who are always with us to provide consultation services and partner with us for webinars and seminars and workshops. Okay, so this slide here is to show you the contact numbers and email addresses if you require any further assistance or for bookings or for inquiries. And whatever that I have mentioned before can be accessible, the information can be accessed at this website here, crlfom@um.edu.my. So if you need any further information, please do visit our website or contact our staff via these numbers. Okay, so that's all from me on the introduction of CRL. And now we will move on to the sharing session. Okay, so our first invited speaker who will be sharing her experience on using our facility is Associate Professor Dr. Tengku Fathlun, Tengku Kamalden. She has been with us since 2012. Back then it was still called MBL, and she started with us when she first came back from overseas to set up a research project at UM. Allow me to give a brief introduction on Dr. Tengku Ain. Dr. Tengku Ain is a clinician scientist and a retinal surgeon at the Department of Ophthalmology, UMMC. She was awarded Doctor of Philosophy in 2013 at the University of Oxford for her work on retinal neuroprotection. So today she will share her experience as one of the users of MBL from the researcher's perspective. So it's my pleasure to invite Dr. Tengku Ain. All to you now.
Thank you very much, Prof. Shatrah. Assalamualaikum warahmatullahi wabarakatuh, and good morning to everyone. First and foremost, I'd like to thank Prof. Shatrah and her team for inviting me to give a talk on today's session, highlighting CRLs or the facilities that is available in Faculty of Medicine and UMMC, and how it has helped me along the way in my research work and research career at the Department of Ophthalmology. So I've got no financial disclosures related to this talk. And so just following up on how what Prof. Shatrah said, brief research intro into my research training background. I am an UM alumnus, both in 2000 and masters as well. And I pursued my PhD for neuroprotection studies in retinal cells and in the UK and Oxford. And I returned to UM from PhD in 2011. And it is during my PhD studies that I was trained into molecular research work and lab-based programs. So my thesis was focused on neuroprotective studies of antioxidants on the retina and retinal ganglion cells at the Nuffield Lab of Ophthalmology. And as I said, I received training in primary and cell line cultures, molecular signaling pathways, and various lab methods, although I do have some clinical experience, but my main focus was lab work. So upon returning, as many of us like to do as researchers or clinicians or not, we like the opportunities to start off our own research focus and topics and find our research niche and look at places and opportunities where we can grow and continue to practice the research skills that we have learned. So typically like that, I look for lab benches and space and facilities, and it just so happens that at about the time that I was looking for spaces and there was grants, and that was when MBL or CRL now has evolved into a shared lab facility, which was a new direction of the Faculty of Medicine. Before that, I considered looking for lab benches and even trying to toy the idea of renovating our own department's small an isolated wet lab rooms as a lab space, but there were many issues that faced us, and it's not a conducive place to start basic science research. You need lab certification, and you need lab safety, you need renovations, you need maintenance, you need skilled manpower to man the lab or manage the lab, and all this comes with funding, which we lack to start off a lab in the department. And so in 2011, 2012, the centralized shared facilities was just opened up, and the bench, it was a new concept then, and the lab benches and lab spaces were up for grabs. And I think I got the last bench at the genomics facility that was still available. The benches are rented out on an annual basis or monthly basis, depending on your grants and your research duration. And there were several, as Prof. Shatrah mentioned, there were many modern equipments and state-of-the-art equipments provided at minimal cost or complimentary that comes with the rental. And the concept of a shared lab facility was something that was similarly done in overseas labs, where they have similar lab equipments for sharing or for rental, centralized genetic facilities. Only thing missing was a vending machine, which I saw in Hopkins, where they had a vending machine of QIAGEN kits that the researchers can just get at any 24 hours, and you don't really have to order and wait for 3 months. But apart from that, everything else was quite similar to the overseas concept. To a young clinician, then young clinician lecturer, and who's opening up or exploring opportunities for research, not having our own lab, it's quite a daunting task for a clinician to start off, to initiate a basic science research. But that's the skills that we get, and it will be wasted if we don't explore and expand. And so the CRL facilities provided a very open and organized concept, very user-friendly, and the concept of sharing facilities and lab equipments. And what most researchers would realize now is that is key, because to start up your own science research, you need a lot of equipments and you need a lot of support. It's almost impossible for you to have a grant just to get this up. So the CRL supports you in every way, without you having to come up with your own equipments, because everything is provided for and everything is maintained, and there is a manager, there is an organized structure. And it's especially useful for clinicians, and it encourages clinicians without their own labs to explore research into lab-based projects. And so it facilitated our work. It broke down barriers, meaning that clinicians do only clinical research. We can also start to do translational research. We can do basic science research. Facilities are up for grabs. The opportunities is more abundant now than they were then. And we can start taking in research students in basic science and lab-based work as well. So we get to expand our research focus. So when we first started in 2011, 2012, or maybe 2013, so we started at the MBL lab, and this was some of my team. It's the location, as Prof. Shatrah said, is at Block B, Level 4, Faculty of Medicine. To my fellow medical alum listening in, I saw some of your names there. To put things into perspective, this is where the anatomical dissection hall was during our medical school days. So the entire dissection hall area and the pathology museum has been converted into a lab space, which you will see in the following pictures. And so you've seen these pictures as well from Prof. Shatrah. This is the genomic facility, where it's much cleaner, purely gene work to minimize contamination. And there's another wing for the protein work. So this is where I'm located. And this is again some duplicate photos from Prof. Shatrah. So this is our pride then, our first lab bench space, my first ever lab bench space. That we purchased small equipments, but then later on we found out that some equipments were duplicate, but it helps because sometimes there will be users sharing our equipments when the usage of the shared equipments is busy or is overbooked, so we can get to share our resources. So this is how we rented the entire bench for several years. You can also rent half the bench. So the concept is good because it's a centralized space. It's a shared facility. You're not alone there, and people do go there just for work, and you don't have to worry about who manages, what's the equipment safety like, who goes in, who goes out, because it's all taken care of. And so some of our activities then using the common equipments, we did PCRs, we centrifuge bloods and for sample analysis, for serum and plasma analysis. We extracted RNA and DNAs, and these were some of my teams on the bench. And so in short, the facilities gave us bench spaces, workstations for students, postdocs, equipments for protein work and genetic works, also allowed us to calibrate pipettes. Sample storage is also important. We were able to store our samples for many years in the -80. It also has meeting rooms and lecture halls, which we use for free for research meetings. And this might sound cliche, but it really helped us to explore just purely research work, non-clinical work, but just purely research work. And this is the kind of support that I found that as a clinician scientist, you really need this. It really supports you to have a location where people that you meet are those are reps from the equipment side or the basic science side. It's a nice fresh of breath air from pure hospital ward clinic setting. So this is a picture of the meeting rooms where we use it very, very frequently, and the lecture halls, which sits, I think, about 50 packs in the non-COVID days. And it's free for use for FOM, especially for research activities. And the other equipments that I think Prof. Shatrah has mentioned. So what are the pros and cons that we found as a user? The main advantage of the MBL is its central location in the Faculty of Medicine. So it facilitates research students, and it also facilitates us to bring samples from the hospital because it's just within walking distance from the clinics and the wards. And so how we do it is we get clinical samples in terms of bloods and other body fluids from UMMC, transported in cooler boxes and walk all the way to the Faculty of Medicine, and then we process in the lab. The door is open at all times during office hours. In the genomic aspect, you do need a card access for the genetic side. However, the major limitations as a user I found, especially as a clinician with limited time for research, is that it is not accessible after hours, and it's not accessible during weekends when I can spare some time doing research or lab work. It's only open during office hours, and as a clinician, I think most clinicians knows you will get tied down with clinical work during office hours. So you need to kind of balance it up and find time to do to have more flexible timing and additional timing at times at the expense of your own personal time to do research. So I found that was my major limitations to do work after clinical work. And also at that point, there were limited cell culture space. So we explored the other arm of MBL, which was the Translational Clinical Lab. And that is located at the top floor, one of the floors for the building for the women and child health in UMMC, and it's just above the wards, the pediatric ward, so it's within the hospital compound itself. It's quite a large place. It has several labs, one or two main labs. These are the shared lab facilities. There are also other departmental labs, but these are the one that is open for research, other research clinician researchers to use. Security and weekend access, it's quite a secure place. The working time is more flexible if you need to work after hours, because sometimes you need to see your cells after specific time frames, and sometimes that extends after hours, or if that's the only time that you get to do work. you can plan your time. All you need to do is get permission from the security downstairs, get a weekend pass, and they'll give you an access card, and then they'll know who's inside. And sometimes there might be security checks. So this is quite standard for at an international level as well. So it's quite good. So we did some of our work there. We shifted some of our projects. And this is a grainy photograph, but that's how it looks like. Yeah, it's divided into a wet lab facility and a dry lab facility, uh, and and that's where we get to do cell cultures. Again, these are all shared equipments and cell cultures for the clean cells and dirty cells. Dirty meaning some cell cultures deals with infective material. So that's what it means. Dirty does not mean contaminated. And there's also a microscope back there for you to assess your cell culture growth. And again, these are some of the facilities. What we did purchase, however, some of the grants that we got was a droplet digital PCR. Then it's a next step generation from qPCR. It's some of you might know it is a mixture between a qPCR. It's a marriage between qPCR meeting flow cytometry. You give you an absolute quantification of your targets. It is there in TCL. I think we've opened it for use on a rental basis. The next step after droplet digital PCR is digital PCR, which I don't think we have it, and that's a bit more costly. The rest of the equipment that is available are more than adequate for you to conduct your research, and that's our bench space, and it's very neatly divided into compartments, so that's your border. So when you work, it kind of gives some sense of discipline to the user and to the students to keep your workspace tight and organized at all times. That's the dry lab facilities where some of the storage spaces, and you can do ELISA and spectrophotometry and qPCR. And as Prof. Shatrah mentioned, some study areas and mycoplasma testing for cell culture work. So this is our current research activities. We use both labs. We analyze human body fluid samples for ophthalmology research in the eye, and we also conduct retinal cell culture research. The common experiments that most clinicians can perform, you can analyze your body fluid samples for inflammatory biomarkers, proteins. You can do ELISA. It's very simple to do. It's not difficult at all. You can do PCRs, ddPCRs. You can do DNA, RNA extractions. You can do protein studies with Western blots. You can also expand into cell culture studies and do immunocytochemistries and so on and so forth. And as a support, I must say this, as a support with CRL, this would not be possible without the support and the facilities and the equipments that have been provided by FOM and CRL. It allowed me to be successful in my grant application for the Global Ophthalmology Awards program in 2018 and 2019. I got another research grant from the Alcon Research Institute, and this is a pretty big grant. And it would not have been possible without the kind of support that we get. So what we have is actually very high international standards. So the key point is CRL provides the adequate and important support for lab-based clinical researchers at UMMC. And this is a huge bonus for us actually in UMMC and FOM. They looked after the equipment maintenance, calibration, also provided even as a simple thing like pipette calibration. There's security access, lab safety is practiced, is of very high standards and maintained. There's always a lab staff available to help. There's always technical support, as you have heard about the many workshops that is being conducted. And also having shared facilities allow researchers from different groups to network and the students to network and talk to each other, and there's a lot of crosstalk. And when in that process, there's a lot of vital information that is being shared for networking and troubleshooting, and this is very, very important as a student. And the advantage to the embarking clinical researchers with all these facilities is that now you can focus your grants on consumables. You do not have to buy or purchase any more equipment. So it's less grant amount required, and hence high chances for you to get your funding application approved. The lab-based research, the location is very strategic. I think it's designed so that it's very accessible to clinicians at UMMC to walk, and and for the students as well. And there's always opportunities to share information between researchers and students, and some students really have good tips on troubleshooting because they do this all the time. And for those embarking clinician researchers, there's a lot of resources around to support your research into translational clinical research at UMMC and FOM. It might not be the same case in other parts of the country, but it is of pretty high standards in UMMC. I've been very fortunate to have experience working in labs in Oxford and Hopkins, and I must say that the shared lab facility is of a very high standard, and it's also very clean. Very little clutter. And starter tips to those trying to expand into translational clinical work. Because of our time constraint and the nature of our job, balancing clinical work, academic work, publication work, and etc. Now putting another mixture of basic science lab or translational work, you need to network so that you minimize your time trying to troubleshoot. You need to collaborate because that's the smartest way forward, because there's always team members that gives you the strength in the expertise. Some are really good in PCR, some are really good in DNA work and biomarkers work. So you need the expertise, and collaborate is the key. And then together you form a teamwork, and that will help you to project your research career forward. So good luck, and I hope that insight helps future researchers. And that's my email if you need to contact me. And special acknowledgements to CRL, specifically to Puan Tiba, who's seen me through 10 years already, and Shatrah and Musfira, my previous students. Thank you very much.
Okay, thank you so much, Professor Dr. Tengku. That was a very enlightening sharing of your experience with us. The second speaker today is a graduate research student, and we think that it will be interesting to hear what our students have to say about the labs and the equipment and the facilities. Today we have Ms. Yogaambikai Paramanantham. Ms. Yoga is a pharmacist who is pursuing her PhD in cancer biology. She is under the supervision of Dr. Nur Akmarina from Faculty of Pharmacy and Associate Professor Dr. Ivy from Faculty of Medicine. She is currently a core user at TCL as well, and today we have invited her to give a talk on an in vitro investigation of chemoresistance in bladder cancer at the Translational Core Laboratory FOM UM. Let's welcome Ms. Yoga.
Thank you, Dr. Shatrah. Please allow me now to share my screen. Okay, can everyone see my screen? So thank you, Dr. Shatrah, for the introduction, and a very good morning, everyone. Please allow me to present my experience as a young researcher at the University of Malaya. And through this short snippet of my talk, I will take you through the brief outline of my study, and then go through a little bit of the equipment that I've used, as well as then I'll take you through a little bit of photos just to wrap it all up. So briefly, the outline of my research focuses on muscle invasive bladder cancer, whereby we hypothesize that the tumor microenvironment promotes tumor progression by making it more invasive and more chemoresistant while at that. So therefore, just give me 1 second, yeah? Okay. So therefore, we proceeded to study this. An in vitro translational study was actually designed incorporating clinical tissue samples with the collaboration of the surgical urology team and cell lines to enable a bench-to-bedside translation outcome. So using harvested conditioned media from the tissue samples, I treated cisplatin naive and cisplatin resistant transitional cell carcinoma cell lines, and then I observed the effects of the TME tumor microenvironment secretions onto the chemoresistant changes. This was also perhaps the main reason as to why I chose to do my research in TCL in the first place, as it was very conducive for me to perform all of my work in a single laboratory. This table is just the gist of the basic experiment workflow that I used to address my study objectives. The columns in blue were the experiments that were conducted, and the adjacent columns in white are the instruments that I've used to enable them, either the instruments or the services provided by TCL, either one. So the workflow begins with cell culture, which uses the cell culture room and its facilities, by which the room sterility is ensured by the staff to maintain contamination. Every clinical sample that we receive as well, and that has been successfully cultured, undergoes a mycoplasma testing using the services by TCL. And then bench work is done to determine the drug IC50 and the gene expression of the RNA. To do so, various equipment at TCL has been used and is listed down here. But these equipments can also be used for various other molecular work as well. For example, the spectrophotometer here reads fluorescence, it reads absorbance, it reads luminescence in multiple modes. You have got the kinetic mode, you have got a plate mode, and all of that. And as you not only can rent the equipment, but you can also engage in the services provided by TCL, by the lab personnel in TCL. It is very conducive for people from both molecular work and clinical backgrounds to carry research, as Prof. Ayn just mentioned. So now I'll take you through snippets of my lab work in just a couple of pictures. TCL has a cell culture facility that has been mentioned earlier by Prof. Shatrah and Prof. Ayn, and that is separated according to the type of tissue that is being cultured. For example, BSC1, which is termed as a clean hood, only sees the work of cell line culture, whereas BSC2, which is termed the dirty hood, only sees the use for clinical samples or primary tissue culture to avoid cross-contamination. And both the cell culture rooms has their own incubators as well, and it is separated into 2 rooms because of the COVID situation currently. So it is really quite safe to do your work. These are snippets of how my clinical samples are processed and cultured. So upon receiving patient consent with the assistance of the urology surgical team, tissue samples are procured during TURP or radical cystect procedures, and then they are transported in ice from the OT to TCL. And these are done in prior labeled tubes, which has culture medium in them. So the tissue samples are then processed, like you can see in figure 3. And the trial images in figure 4 are examples of how the tissues are cleaned, then they are minced and digested using collagenase and further cultured down prior being isolated to the specific cells that we want. The different freezers in TCL allows for cells and tissues to be maintained and stored for long term. I personally cryo-freeze my cells in the -150-degree freezer upon propagating them. And then to grow them back, I speed thaw them, as you can see in figure 6, in a water bath. And then I spin them down using a refrigerated centrifuge. Figure 8 is an image of the cultured translational cell carcinoma cell line, 2 passages upon being thawed. So amongst the many equipments at TCL, one of the main equipments that I use for a lot of molecular work is a multimode spectrophotometer, especially used for my colorimetric assays. So figure 9 is an example whereby I performed the MTT assay using the multimode spectrometer. And the MTT assay is an assay to observe cell proliferation, and then it's further quantified down according to the difference in absorbance. For example, when you see the yellow turn to purple, the changes in the formosome crystal formation, that absorbance would give you the reading of the cell proliferation. Another assay which I use for the multimode spectrometer is the Bradford assay, which is used to quantify protein in the conditioned media that I mentioned earlier. So moving on, ELISAs, which are used to measure cytokine activity, as mentioned by Prof. Ayn earlier, in this study is performed using clinical samples, and the fluorescence reading is done using that spectrometer, as I mentioned earlier. TCL equally provides the service of running this ELISA for you for both internal and external customers. To then perform downstream genomic work, RNA is extracted and the fume hood is widely used. So the fume hood is not just used for the RNA extraction, but for any work or any work really that involves chemicals or toxic reagents or flammables or organic solvents, and to maintain safety. And then once the RNA is extracted using a two-step RT-qPCR method, the cDNA is first synthesized using a thermocycler, which is an equipment that has thermal blocks and discrete holes in them. And that allows a series of pre-programmed steps to produce a cDNA template prior being used for RT-qPCR in the step 1BA equipment. So figure 14 is an example of the raw qPCR output of CT values from the RNA that I've just analyzed. So last but not the least, TCL has also a fluorescent microscope, which has live imaging features to enable live microscopy observations. So figure 15 is an example of its implementation using the phase contrast, which I use to measure cell migration using the wound healing assay. This microscope has also UV features, which allows for fluorescent cytochemistry and using various things like DAPI, FITC, trypsin, all of that. So that is all from me as a budding researcher, and I'm very grateful for the assistance rendered to me by all of the CRL staff and team. Thank you very much.
Okay, thank you very much, Ms. Yoga. Thank you for sharing all the different equipments that you have used and sharing the what sort of results is expected from the use of these equipments. And we hope that the researchers at FOM has some idea or is enlightened towards the kind of experiments they can venture into, having known that these equipments are available at our facility. We move on to the Q&A. There's one question from Chan Yu Quen. Various researchers are trained to different standards of practice with equipment. How does the center ensure that the users comply with acceptable standards and keep risk to other users low or non-existent, especially dealing with body fluids? This is especially out of the office hours when support staff are not around. Okay, this is a very good question. Simply said, whenever we have new users, they will have to be trained to use the equipment several times. The user will be observed while they handle the equipment. So there's a training, there's monitoring before we can allow them to use the equipment on their own. We have standard SOPs for each and every equipment. So if the users comply to these SOPs, we hope that we will minimize any risk to others. If with regards to the dealing with body fluids, so far we do not accept, if you're talking about infectious samples, we don't accept as infectious samples. Before you start your experiments, you will need to declare the kind of samples that you deal with. For our facilities, just to inform you that for the kind of experiments that we do at our facilities, the cellular work, the molecular work, these are usually done under sterile conditions. So we use disposable sterile equipments and disposable apparatus. So it's quite difficult to get any cross-contaminations and so on. Back to the use of equipment outside the office hours. Just now, Dr. Tengku, I mentioned about not being able to access the MBL outside office hours. So once upon a time, that might have been the ruling, but now we have become a bit more flexible. So we do allow access outside the office hours, but it's a case-to-case basis. So you will have to fill in a form to request so that we have a staff with you if necessary. But if you are already a seasoned user or regular user and you have a bench space with us for months and we think that you can handle the equipment on your own, so we might allow you to work without the support staff around. Cancer samples from, okay, so next question, I hope that answers you. Cancer samples from patients may contain virus. How do you ensure this is safe? Okay, again, this is back to the my answer just now that we don't accept infectious samples. But if you do have samples, cancer samples containing viruses, we will have to, if there's a risk of this, then I would suggest that we would suggest that the users try to be extra careful.
I think Prof. Shatrah is muted.
Yes, I think so too.
I don't know if I can answer this because I don't work on cancer samples with virus cells, Prof. Chan. But I hope that Prof. Shatrah's answers to a certain degree can answer the question that you seek. Do you have experience about this, Yoga? I mean, do you work in the T-cell culture rooms?
Yeah, so I do get bladder cancer samples from the bladder tumor patients. And what I do to maintain its sterility is that I wash them with various, antibacterial stuff. So I wash them with pen strap and then antimicrobials and stuff like that. And also we perform a mycoplasma detection on them so that I know that it's not carrying any sort of mycoplasma.
Yeah,
that's the most I can do.
Yeah, I agree. Because I think most of us, I think, would have been orientated as users at the lab to follow the strict SOPs, and I think they know that, so they're quite strict in who are the users, and that the users are trained. You can't just open up to any user without the training. So they are as a user, you kind of have a registry by the admin, and then they would know the kind of samples that you would use, and let's say there's always a mycoplasma testing, because whenever there's multiple users, we all have been trained to wipe down and wipe. Be OCD and every lab rat is an OCD. So we I think that kind of we kind of protect each other in that sense for the next one. Prof. Shatrah is still not on the line, so I'll try this as much as I can. Prof. Chan has another question. Some virus with no treatment facilities may need level 4. As far as I know, TCL is not classified to receive virus samples up to level 4, right? So I think that's when you need BSL. I think you need BSL 3, right? And BSL 4. I think those facilities are maybe available in TITREC, because TITREC has the highest level for research in terms of virus, including COVID. TCL is not prepared for or accredited for that, so it's just I think just the basic 1 and 2, and we need for the routine non-infectious low risk research.
Because our BSCs are also just equipped with just basic HEPA filters. It's not not anything else.
So for that, Prof. Chan, I think TITREC has those facilities for very high level virus infectious materials. Okay, so thank you for your questions, Prof. Chan, and your interest. And Prof. Ng has already also mentioned could consider using ultraviolet C to deactivate the viruses. And as that's quite routine, we use the ultraviolet light on the cell culture before we start, and we deactivate all the pipettes and the tips before when we do sterile work. So it's quite a high standard, I must say, and it's clean than some of the other labs that I've been to. I think Prof. Shatrah is still. Prof. Shatrah, please take over.
I'm sorry, there was an
Sorry.
issue with my audio just now. Thank you for covering up for me. Okay, so I hope that answers your question. We hope that the samples that you bring in do not contain any infectious diseases, because we don't handle that, as mentioned by Prof. Ain just now. If you do have samples like this, I'm sure there are other divisions that can help you out on this. And if there's another comment from Prof. Chan, I like the idea that we have to protect each other. However, we must remember that honesty is all about here. Honesty is what you do not do when others are not around. Totally agree. This is part of being to have this poise within us. Okay. So you have to have the integrity. Okay, so we are coming close to 9 o'clock. If there's no other questions and comments, I would like to thank the breakfast at UMHealth administrator, Dr. Shams, and the UMHealth unit, and our two invited speaker, Professor Dr. Tengku and Ms. Yoga. Thank you so much for coming and joining with us to share your experience. If you do have any further questions, do not hesitate to contact us. You can either contact myself or any of our managers or science officers, or you can easily visit our website to look through the facilities that we have, the services that we have, and do visit our research card 360 as well. So if you need to purchase anything, this can be accessible within 1 or 2 days, compared to 2 to 4 weeks if you buy from a third party. So that's all from us. Thank you so much on behalf of CRL and the team. I would like to thank everyone. Have a good day ahead. Thank you.
Thank you very much.
Thank you. Thank you.